Hi guys, a very warm welcome to all of you in our Gurmantra Academy and your dreams are our visions here. So today it's your practice session. So we are going to practice session. So we are going to practice some question. It is going to be like your test. So today it is your test. I hope you guys have prepared and come for the test. So I'll tell you some of the rules before we start the quiz. The first rule is that you will have 30 questions, each of which is 2 marks, so totally 60 marks. So this test is for 60 marks. Second, think that it is like your gate exam. Okay, think that it is your gate BT exam itself. Okay. Third one, for every question 2 marks. Okay, so for correct answer you will give yourself 2 marks, self evaluation only, no. Then for the wrong answer you will give minus 1 mark. Finally, when the quiz is over, you will total the marks and tell me the total. Tell me the total. Okay, tell me the total. Okay, so when the test is over. So this is what is your task for today. Let's start with our test. All the very best, guys. Keep doing well. Have a notebook with you. Okay, suggest to have a notebook like this. There you can actually write question number 1, 2, 3 till 30. Till 30 you can write, and you can give marks here. You can give marks here, like for example 2, 2, 2 or minus 1, 2. Like this you can give your marks, and finally you can see the total. Finally you can see the total. Calculate the total, whatever your marks you're getting. Okay, out of 60. Out of 60, how much you're getting? This you need to let me know. Okay, so this is the task for today. I hope you guys are doing your homework questions which I'm giving you every day, right? Because it is for your own purpose. Initially it was planned so that you can understand how much you have actually learned. It's like a recall purpose. So I hope you're doing it well. Okay, so let's start our quiz for today. So this is your question number 1. Do not look for this one as your question number. It is actually the question number from which I have taken. This is the source. So this is basically the question number. So it's the first question. Read the question carefully, understand it, answer it. Write it in your notebook whether it is A, B or C or D. We will discuss the answer after 1 minute is over. Okay. So this is a very simple question. You don't need much of explanation for this. So which of the following is not called a cell? It is basically your virus. Okay, because virus is considered to be a non-living particle. Next question. So if your answer is correct, give yourselves 2 marks. If it is not, minus 1 marks. Finally, total. Okay. So what are microfilaments composed of? So in cytoskeleton we studied about microfilaments, intermediary filaments, microtubules. I hope you guys remember. So that will answer this question. Okay, so the correct answer for this question is your option B, actin. Okay, actin comes under microfilaments. Microtubules will come under this tubulin. Okay, next question. Lysosomes are known as suicidal bags because what property of lysosome makes them as a suicidal bags? I wanted to let you know, guys, that there is a group called as Gurmantra Academy in Telegram. Okay, so you can join them for discussion along with your peers. Okay, you will get all the updates about the classes, quiz, sessions, everything. Okay, so lysosomes are known as suicidal bags because they have hydrolytic activity. Next question. The function of the centrosome is. So, what does centrosome actually consist of? It consists of centrioles. And where is the function of centriole? That will give you the answer. This the correct answer for this question is your option A, spindle fiber formation. Next question. Which cell organelle is involved in apoptosis? We studied this just yesterday. The apoptotic bodies, where will it go? You know what? These questions are making me tempted that I want to answer them immediately. But the thing is, I'm the educator here, so I can't answer them. But it's really tempting. So I hope it is as much as tempting for you guys to answer the correct answer. Because I think quiz sessions are more fun than the lecture. Because lecture, yes, it is long, it is lengthy, it takes patience, but quiz is fun. So the correct answer for this question, which organelle is involved in apoptosis, is your lysosome and mitochondria. Okay, mitochondria is also involved and lysosome is also involved. Okay, apoptotic bodies will go inside the lysosome. The the intrinsic pathway will be a part of mitochondria. So both will be the correct answer. Multiple select questions. So the enzyme of the TCA cycle reside in the mitochondrial membrane. Is it true or not? So where does TCA cycle takes place? TCA cycle is nothing but your Krebs cycle. Where does it occur? It occurs in the mitochondrial matrix. It occurs in the mitochondrial matrix. So they're asking whether the enzymes for this particular cycle, whether it is present in the mitochondrial membrane. Very simple. You just have to answer it. True or false. Okay, so the correct answer for this question is your option B. No, false. Okay, so all the enzymes of TCA cycle is present in the matrix. For electron transport chain only it is present in your mitochondrial membrane. It is all present in your matrix. Next question. In the TCA cycle, which of the following combines with acetyl-CoA to form a 6-carbon compound? Acetyl-CoA is a 2-carbon compound, so it needs to bind with another carbon compound which is 4-carbon. Only then you will get a 6-carbon compound. So what about glucose? Glucose is a 6-carbon compound initially, but if it combines with acetyl-CoA, it will become 8-carbon, so no. What about pyruvate? Pyruvate is a 3-carbon compound, isn't it? It's a 3-carbon compound. So will it form? Think about it and answer me. So questions like this can be tricky because you need to know how much carbon is present in acetyl-CoA. Only then you will be able to see. To form 6-carbon compound, you need only a 4-carbon compound, and what is that? It's your oxaloacetate. Okay, it's your oxaloacetate. Next question. The meiotic division takes place in meiosis. Where does it take place? Very simple question. This question and the answer for this you have been studying from 6th standard onwards. So you should be able to answer it like in a blink. The meiotic division takes place where? Do you need 1 minute for this? Really? You need. All of you have answered this question already, isn't it? But let me give you 1 minute for those who are not able to answer. Let them think and answer. So there you go. Your correct answer is your reproductive cells, germ cells. Next question. So you need to name the event where the paternal and maternal chromosomes are exchanged during the cell division. What is it called as? The event, not the place. Okay, remember the question. I mean, understand the question. They're asking you the event. Okay, so the correct answer for this question is crossing over. Next question. Synapses takes place between where? Whether is it a spindle fiber and a chromosome, I mean your centrosome, mRNA and ribosome, a female and a male gamete, two homologous chromosomes. So where does this synapses takes place? They're asking you the location. Answer it correctly. Think about it. Take your time. You have time. 15 more seconds. 10 more seconds. Okay, so the correct answer for this question is your two homologous chromosomes. Next question. The stage of prophase 1 wherein crossing over take place. In which stage in prophase 1 your crossing over takes place? Very simple. We are studying this. We are studying this. And you should be thorough with this right now. So, with this 11 questions are getting over. So, 11 into 2 marks, 22 marks is over. So, out of 22 marks, how much have you done? Okay. I hope you guys are calculating, right? For the correct answer, 2 marks. Wrong answer, minus 1 mark. Don't forget that. Okay. So, you will not able to track your progress. Studying, taking test is important as long as you are tracking the progress. That is also important, right? So the stage of prophase 1 wherein crossing over takes place is your what? Pachytene stage. Next question. Meiosis 1 is reductional division and meiosis 2 is equational division because of what? What is different? So the correct answer for this question is your option A. Separation of chromatids is happening. That is why 1 is called as reductional division, 2 is called as equational division. Okay. Next question. DNA synthesis takes place during what phase? I'm not going to give you 1 minute for this. Okay. I want immediate quick answer. Immediate quick answer. In what phase your DNA is synthesized? It's very simple. In the synthesis phase. And it starts from... It starts from your S word. So S phase is the correct answer. Next question. Formation of cell plate starts at. When does the cell plate formation begins? So you need to just remember what are the events in the cell cycle. You will have G1 phase, then you will have S phase, then you will have G2 phase, then you will have mitosis. In mitosis, what are the phases present? Prophase, metaphase, anaphase, telophase. Then what happens? Cytokinesis. So this is your cell cycle. Now remember this and answer the question. Do you know why I'm telling you this? Because answer is present in this. I'm telling you this because when you do not know the exact answer, this is how you can recall your answer. So this is the recall mechanism. Okay, recall mechanism. I'm teaching you how you can recall your correct answer. So the correct answer for this question is very simple. It's your telophase. Next question. Chromosomes are separated during... Very easy. Chromosomes are separated during what phase? Five seconds more, guys. Okay, so the correct answer for this question is chromosomes are separated during your anaphase. Okay, next question. This is from your first unit, biochemistry, but I think you'll be able to answer this because we've already given the test, right? Practice session 1, we have given the test. So you should be able to answer this question. So this is an enzyme scheme that basically depicts what kind of your inhibition. Can you see? The inhibitor is binding only to your enzyme, right? So what kind of inhibition is that? Okay, so the correct answer for this particular equation is your competitive. It's a competitive inhibition. Okay, option D is the correct answer. Next question. G protein coupled receptors exhibit which of the following characteristic? So option 1 is tyrosine kinase activity, is it? ATPase activity. 7 transmembrane domain, nuclear localization, dimerization. So for these type of questions, if you do not remember the correct answer, you can eliminate the options. Use the eliminate the options method. So tyrosine kinase activity is definitely not present in GPCR, so no. Dimerization, no. Nuclear localization, is it happening? Nuclear localization happens in the case when it is a nuclear receptor. So no. ATPase activity. Does ATPase activity exist? You have to think about it. So think about it. No. So the correct answer is your transmembrane. It is a serpentine receptor. It will be like this. 1, 2, 3, 4, 5, 6, 7. Okay. So serpentine receptors will be like this. Next question. Name the most important lipid component of the cell membrane. Cell membrane is present, it is made up of which important lipid? Abundance. When it is asked about most important, think about abundance. Oh God. I'm so sorry. Without my knowledge, I actually pressed the answer. But okay, I hope you guys know it. It is nothing but your phospholipid. Okay, it's your phospholipid. Next question. I need to keep my hands away from the keyboard. By mistake, I'm pressing it. What are the monomers of protein? Protein's monomers is called as very simple question, very easy question. So the correct answer for this question, what are the monomers of proteins is, it's your amino acids. Next question. Fats are made up of what? Very easy, right? Very easy. Fats are made up of, what is it consisting of? Option A, glycerol and amino acids. Option B is your cholesterol and fatty acids. Option C is your glycerol and keto acids. Option D is your cholesterol and amino acids. And option E is your glycerol and fatty acids. So which of the following is correct? So the correct answer for this question is your glycerol and fatty acid. Okay, next question. How many classes of enzymes are there? सोचना भी मत, बस like this you need to answer the question. How many classes of enzymes are there? Okay, so you know the answer for this question, but I want you to name also. Okay, naming them is also important. Enzyme classes. So in your notebook, write the names also. Write the names of the enzyme classification of enzyme. The correct answer is six. Six classes of enzymes are there. Next question. A competitive inhibitor of an enzyme, what does it do? Very easy, right? A competitive inhibitor of an enzyme, what does it do to your Km and Vmax? So when you just see a question like this, immediately draw the graph. Immediately draw the graph. Okay, draw the graph. So Vmax will come somewhere here, Km will come somewhere here. Okay, so like this and like this. Can you see Vmax is same, Km is different. Km is different. And how is it different? That you need to tell me. Okay, how is it different? Km will basically increase and Vmax will be same. Next question. Km equals to the substrate concentration when the reaction rate, when they are asking about V, what is the value of Vmax at Km? Km is the concentration of the substrate when the reaction rate is what? So this is what they are asking. This is the correct answer for this question is half Vmax. Okay, next question. It actively synthesized rRNA. So basically they are asking where is rRNA synthesized? The question is not put correctly. Where is your rRNA synthesized? That is the question. Whether it is present in your nucleoplasm, nucleolus, nucleus or all the above. rRNA synthesis takes place where? Okay, so the correct answer for the question rRNA is synthesized in is your nucleolus. Next question. Which organelle possess hydrolase enzyme? Very simple, guys. You should be able to answer this immediately. You don't even need 1 minute. Okay. So the correct answer for this question is your lysosome. Next question. Select the correct option from column 1 and column 2. So one of this question we have done, the first question's answer is present in this match. So we have already seen microfilm. I mean, I'm not telling. Okay, so do it by yourself. It's easy. That's what I'm saying. Try to eliminate the options. Very easy, isn't it? Just by your common sense and the knowledge which you have gained from my class is more than enough to solve questions like this. Okay, so the correct answer for this question is microfilaments. Where does it belong to? Actin. So P2. P2 means this option is eliminated. Microtubules, tubulin. Q3. Q3 means this option is eliminated. Flagella, flagellin. R4. This option is eliminated. Outermost layer of the bacteria is glycocalyx. S1. Okay. It is basically S1. So here it should be S1. Only then it will be correct. Okay, S1. So I think this is given as wrong, but it's okay. Let's leave the option. Let's write here. Microfilaments. It is for actin. Microtubules. It's for tubulin. Flagella. Flagellin. Outermost layer of the bacteria, glycocalyx. So this is your correct answer. Okay, correct match. Next question. In column 1, organelle, and in column 2, its function is given. You need to just choose the correct option. Very simple. So we have already studied lysosome is your suicidal bag. Correct? So R1. R should be 1. That actually gives the correct answer. So if R is 1, this is wrong because here R is 4, R is 2 and R is 3. Just with one answer, just with one match, I've solved this question. Okay, I'm just telling you how you can solve the question. Do you understand? Okay, so chloroplast is for photosynthesis. Easy. Smooth endoplasmic reticulum is for your steroid synthesis, mitochondria, ATP. So this is your correct answer. Next question. So in the above signaling cascade, which one of the following molecule is denoted by B? It's basically your JAK-STAT pathway. Okay, it's a JAK-STAT pathway. So the ligand is binding to the receptor, dimerization of receptor, autophosphorylation. Then your adapter proteins will come and bind. So what is that adapter protein B? That is what you have to answer. So the correct answer for this question, where what is the B, is your option D. That is your SMAD4. A will be your SMAD 2 or 3. B will be your SMAD4. Okay. Next question. Which of the following is not a secondary messenger? So cyclic GMP they have given. The diacyl glycerol they have given. Inositol IP3 they have given. Phosphatidyl inositol they have given. Okay, phosphatidyl inositol. So which of this is not a secondary messenger? I've told you the answer itself, isn't it? So phosphatidyl inositol is not a secondary messenger. Next question. So this is your last question. The enzyme that catalyzes the splitting of PIP2 into two molecules, that is IP3 and DAG, is, so PIP2 is split into IP3 plus DAG. So what is the enzyme used here? That is what they are asking. Okay, that is what they are asking. What is the what is the enzyme used here? So the correct answer for this question is what? Phospholipase C. Okay, phospholipase C. This is your correct answer. Okay, that's it, guys. Your test is over. What you have to do is total the marks and tell me. Okay. So if you like my lectures and if you want your friends also to take the benefit of it, ask them to join the GATE biotechnology courses where you get both biotechnology as well as Excel courses. Okay, both will be present in one particular single package, and you can use code Saranya 10. Okay, code Saranya 10 to get 10 percentage off. Okay, so thank you so much, guys. Have a nice day. Keep preparing well.